The three-dimensional computer graphic drawings of plant and animal cells might look quite nice, but a little bit impractical when answering examination questions, because you really want to draw a three-dimensional drawing of a plant or an animal cell and label it during an exam session. That's a little bit complicated unless you're pretty good in arts and in drawing. So what I'm going to do now is I'm going to draw you a two-dimensional cross-section of an animal cell and of a plant cell. Remember that every time when you draw something, you also have to label it. Now, I'm not going to do that. But you should label it in your notebook. I'm not going to do that for practical purposes and for time purposes. I'm going to start off the animal cell with the cell membrane. So I'm drawing over here the cell membrane. In your notebook, you add a line and you write down the cell membrane or plasma membrane. It's the same thing. Every line that I draw represents a membrane. And I always start off with the cell membrane, because this is the structure which defines the borders of the cell. The cell membrane, I'm just going to draw a line here, but you write down cell membrane. The cell membrane surrounds the cytoplasm, which is the liquid content inside the cell. Inside the cytoplasm, you have a very large cell organelle, and of course you know that this is the nucleus, and the line over here you can say nuclear membranes. It's plural, so there should be a second one, or you can just say the nucleus. So, and when I say it's plural and there are two of them, I have to draw two membranes very closely together. Not so easy on my graphics tablet over here. You should have an easier time in your notebook. So some people also refer to this as a so-called nuclear envelope, because there are two membranes. Now, the inside of the nucleus is also filled with a liquid which contains the DNA, and this liquid content is referred to as the nucleoplasm. The nucleoplasm. And inside here we have the DNA. Okay, that's the chromatin, that's the DNA. And there's also a region which is a little bit darker. And I'm not going to draw a circle now, because if I draw a circle, I'm drawing a line, and this would imply a membrane, but it's not a membrane. Rather, it is a region which shows up on electron micrographs, which is a little bit darker. And this is the nucleolus. I know these terms all sound very similar. Nucleoplasm, nuclear envelopes, nucleus, nucleolus. Okay, so that is over here, the nucleolus, and this is the place where RNA is being synthesized. More on that in a different video. So that is basic. Yeah. Before I forget, many students they draw the nucleus far too small. Do not forget, it's the biggest cell organelle, and some people draw it as a small circle somewhere, which is strictly speaking not correct. Now, the outer membrane of the nucleus has outfoldings, and these outfoldings they kind of follow a little bit the outline and the shape of the nucleus. And am I drawing a line here? Of course I am. So this is also made of a membrane. It goes back here again. Some of these are also detached a little bit and goes like this. And that's the rough endoplasmic reticulum. And it's called rough because on the surface of these this membrane, we have these small dots here, and these small dots are ribosomes, which are found on the RER. RER is an abbreviation for the rough endoplasmic reticulum. So over here, you would probably want to label rough endoplasmic reticulum, RER, and these dots over here are ribosomes. you label ribosomes over here, which can be found on the RER, and these ribosomes make the RER appear rough. And if there's an RER, there must of course also be somewhere a so-called an SER, which is the smooth endoplasmic reticulum. And I think I do not need to say that they do not contain any ribosomes. Again, I'm drawing a line over here, so I'm drawing again a membrane structure, and that's the smooth endoplasmic reticulum without the ribosomes. So I'm just going to continue drawing some ribosomes over here, just to make this a little more clear. It should be connected, of course, to the RER, not just floating around. So that's the smooth ER, that's the rough ER, ribosomes found on the rough endoplasmic reticulum. And we move on over here. There are inside the cytoplasm a few other interesting cell organelles as well, and one of them would be the so-called the Golgi body, and the Golgi body looks like a stack of discs. I'm just going to draw it like this. They're kind of stacked quite tightly. And this is the Golgi body. You write it with a capital G because Golgi, that's named according to a scientist. And Golgi is the name of the person that's named, who discovered it. It's the Golgi body made of membranes. And surrounding the Golgi body or Golgi apparatus, you have these little bubbles, which we do not call bubbles, but we call them vesicles. And these little bubbles, no, you call them vesicles, I'm drawing a line, so they're also made of a membrane. So these are the vesicles, and these vesicles, they form from the RER and they travel over to the Golgi body. That is explained also in a different video. Over here we're just concerned about the structure of an animal cell. So the next thing that I want to talk about is another cell organelle. It's the mitochondrion. These bean-shaped structures. I think I do not need to say it anymore. This is a membrane, and there are infoldings. Please draw them nicely. I will show you a drawing of what some people have drawn, which is not so nice. That was not so nice. Okay, like this. It's also a membrane, of course, the inner membrane of the mitochondrion and the outer membrane of the mitochondrion. The inner membrane is folded inwards to increase the surface area. So you would like to label this as mitochondrion. Do not draw it like this. Just show you. Some people draw the mitochondrion and they draw it like, that's the inner membrane. No, please don't do that. Okay. Also, the Golgi, some people draw the Golgi like this. No, that's not a stack of discs. That's not nice. Okay, draw it properly. You also have inside an animal cell, besides these vesicles, you have also slightly larger membrane-bound bubbles, and these are the vacuoles. But they're not too large. They're not too large. So several of these are floating around here. Food they might store in here. They might store some food. So these are food vacuoles. Is there anything else I forgot? Yeah, of course. You have ribosomes on the RER, and there are also free ribosomes floating around in the cytoplasm. So these are also ribosomes, and they're free ribosomes in the cytoplasm. And I'm just wondering if I forgot anything. There are many other structures in animal cells as well, like, for example, centrioles. Centrioles are a little bit difficult to draw because they are protein structures which are arranged perpendicular. They are little short tubes. And another one is arranged in 90 degrees. And together they make the so-called centriole or centrosome. And they make spindle fibers important for cell division. So, but yeah, I would say this should be pretty much it with the animal cell. In a real cell, there are many more structures, but for right now, I think this covers all the relevant aspects. So that's pretty much it for this one here.